There are two types of nuclear transport, the nuclear import and the nuclear export, and there are specific signals which are needed for these transports. The signal which is needed for the nuclear import is known as the nuclear localization signal. It contains two basic amino acids, lysine and arginine as the repeats. And in the case of nuclear export, the signal is known as NES or the nuclear export signal. And the nuclear export signal contains the continuous stretch of leucine. Cardioferins are the class of protein which actually recognize these signals, and there are two types of them, an importin in the case of the import and exportin in the case of the export. First, let's talk about the nuclear import. When a cargo protein with the nuclear localization signal comes in the cytoplasm, it actually binds with the importin, and then the importin and the cargo protein can enter through the nuclear pore complex into the nucleus. And when this complex gets into the nucleus, it binds with RAN-GTP. So, as our importin cargo complex binds with the RAN-GTP, it actually causes structural change and causes the release of the cargo protein. Thus, the importin and the RAN-GTP are combined, and the importin RAN-GTP complex is now ready to be shipped back into the cytoplasm. When this RAN-GTP importin complex is taken into the cytoplasm, inside the cytoplasm there is a gap, which actually means the GTP is activating protein. So this protein actually activates the GTP's activity of the RAN, and hence the RAN-GTP becomes the RAN-GDP. And that's what happens. The RAN actually dissociates from the importin, and importin is now free in the cytoplasm. The RAN-GDP is taken from the cytoplasm to the nucleus. Inside the nucleus, there is another thing which is known as a GEF. A GEF actually means a guanine exchange factor. Guanine exchange factor actually swaps the GDP and switches it with a GTP, and hence the RAN-GTP is again fully active. It's time for the nuclear export. In the case of the nuclear export, the cargo is inside the nucleus and we need to transfer it from the nucleus to the cytoplasm. So the exportin actually binds to the NES and recognizes the cargo. As you have seen in the case of importin, anything that goes out of the nucleus is combined with the RAN-GTP. The cargo, the RAN-GTP and the exportin is actually combined, and it is actually taken out of the nucleus. When it reaches the cytoplasm, the gap is activated. So the RAN-GTP is converted to the RAN-GDP. Hence, it causes the dissociation of this complex. Then what happens? The RAN and the exportin is actually taken into the nucleus. Inside the nucleus, the RAN-GDP is converted to the RAN-GTP by the GEF activity. And hence, the RAN-GTP and the exportin is ready for the next export. We have studied a lot of things about the nucleus, and it's time for us to explore the cell organelles. And that's gonna be the topic of your next video. See you in the next one.